So, mitosis and meiosis. Shh. Dina, turn around. Okay, mitosis and meiosis is the process in which cells replicate. Okay, so that's what we're going to do. Outline the process of replication, compare mitosis and meiosis, and describe the process of transcription. All right, so DNA, genes, and chromosomes. DNA, we've talked about already, deoxyribonucleic acid. It carries the genetic information for the body's cells. DNA is made up of 4 similar chemicals, like we've talked about. They're called bases, and we abbreviate them A, T, C, and G. They are repeated over and over in pairs. A gene is a distinct portion of a cell's DNA. Genes are coded instructions for making everything in the body needs, especially proteins. Human beings have something like 25,000 genes. Researchers have discovered what some genes do and have found some that are associated with disorders, such as cystic fibrosis or Huntington's disease. There are, though, many genes whose functions are still unknown. Some genes do nothing. Some genes are what is called just junk DNA. Genes are packaged in bundles called chromosomes. Has anyone ever heard of chromosomes? Okay, so humans have 23 pairs of chromosomes. Taylor, put your phone away. If I have to speak to anyone about phones today, I'm having a super not great day, guys. I really don't want to have to talk to anyone about phones today, okay? So please just leave them in your pockets. So total of 46, you get 23 chromosomes from each one of your parents. One of those pairs are called your sex chromosomes. They determine whether you're a male or female, such as some of your body characteristics. So if you're female, you have an X and an X chromosome. If you're male, you have an X and a Y chromosome. So your sex is basically determined by your dad. Okay, so everyone got one X from their mom. And then if you're a girl, you got the X from your dad. If you're a boy, you got the Y from your dad. Okay? And then 22 pairs of autosomal chromosomes, which determine the rest of what your body does. Okay, DNA replication. We've kind of talked about this already. We're just going to go through it quickly. Again, that's why there's no blanks for these few slides. So, remember, an enzyme called DNAgrase makes a nick in the double helix, and each side separates. An enzyme called helicase unwinds the portion of the double-stranded DNA in an area called the replication fork. Several small proteins called single-strand binding proteins temporarily bind to each side to keep them separated. An enzyme called DNA polymerase walks down the DNA strand and adds new nucleotides to each strand. The nucleotides pair with complementary nucleotides on the existing strand. So the DNAgrase walks down and adds the A, T, C, or G to its complementary A, T, C, or G. So if it finds an A, puts a T. If it finds a T, puts an A. Okay? Goes down there. Basically what's happening is they're making 2 new strands of DNA. So each new strand has one old strand and then one new strand. A subunit of the DNA polymerase proofreads the new DNA. So it's on the back, and as it's going down, it's just making sure that everything's matching up properly. A new enzyme called DNA ligase seals up the fragments into one continuous strand, and then new copies automatically wind up into that double helix shape. Building proteins. So remember, it's like building a house. Okay, so the master blueprint is your DNA, which contains all of the information to build the new protein or the house. The working copy of the master blueprint is messenger RNA or mRNA, which copies the DNA. So it copies that master blueprint into the working copy. The construction site is the cytoplasm in prokaryotic cells and the endoplasmic reticulum in eukaryotic cells. The building materials are those amino acids. Construction workers are ribosomes and transfer RNA molecules. And then this is done in 2 steps: RNA transcription and translation. So transcription is the copying of the master blueprint to the working blueprint. The transcription is performed by an enzyme called RNA polymerase and goes through the process. The RNA polymerase binds with the DNA strand at a specific sequence called the gene promoter. Unwinds and unlinks the 2 strands of DNA, uses one strand as a template, matches the nucleotides, so the G's and the C's, the A's and the U's. Remember, RNA has a U instead of a T. Binds the new RNA nucleotides together to form a complementary copy of the DNA strands, and it stops when it encounters that termination sequence. And then translation is where the construction workers take that blueprint and create a protein, build a house. So the mRNA strands move out of the nucleus into the cytoplasm and attaches to a ribosome. Transfer RNA anticodons are a series of 3 nucleotides which match or pair with the first codon on the mRNA strand. The transfer RNA anticodon matches with pairs, and the second anticodon on the mRNA. The amino acid on the other side of the transfer RNA binds. The first transfer RNA leaves to go pick up another amino acid, and the process continues all the way down the mRNA. Okay, and it builds up that protein. All right, now on to new stuff. So,
Can I ask a question?
at the end of my lesson, please. Okay? See, that's right. Okay, so mitosis is the division of somatic or body cells, a process by which eukaryotic cells divide the genetic material into its nucleus, and then, or divides the genetic material in the nucleus into 2 new identical nuclei. Okay, so everything, all the little pictures that we're going to look at are just the nucleus. Okay, they're not the whole cell. The whole cell does divide eventually, but this is just the division of the nucleus. Okay, and it's going to divide it into 2 identical nuclei. Okay? All right. So, interphase. This is when the cell is growing. Okay, the cell grows, carries out its normal processes. It's just kind of chilling. Okay, the chromosomes in the nucleus form a mass of thread-like structures called chromatin, which are composed of DNA and proteins. So they're just kind of floating, hanging out. The replication of the chromosomes during the interphase results in pairs of sister chromatin, which usually contain exactly the same genes at the same loci, or same location. So basically, it's just replicating exactly the DNA that's there. Okay, if you were to look at a cell that was in interphase, you wouldn't be able to see anything. Okay, everything's kind of long and stretched out and spread out really nicely. Okay, you're going to need to know the exact order of which these stages happen and kind of what a generic cell will look like. Okay, so interphase starts it off. Prophase is where your chromosomes pair up. That's how you remember what happens in prophase. You're going to have to memorize the order, but then P for prophase, they pair up. So the chromosomes thicken and shorten, and they become visible under a microscope. The 2 chromatids are joined by a centromere. Okay, so I'm just going to flip back really quickly. Okay, so if you notice, I have individual cells, or individual pieces of DNA just floating around. Okay, when I get here, they've paired themselves up. Okay, the centrioles move to the opposite poles of the nuclei. The nucleolus disappears, and the nuclear membrane disintegrates. Okay, so this white part inside there, that's the nucleus. The nuclear membrane has disappeared, so now it's kind of just floating, just hanging out. And there are centrioles that have moved to the opposite poles of the nucleus. So they've kind of come to hang out over here. Okay, everyone good? All right, so metaphase. The chromosomes meet in the middle. So they find that middle line in the nucleus, and they line up along it, along the equator of the cell. Each becomes attached to a spindle fiber from the centromeres. So remember, the centromeres came out to the side here, and then they've dispersed fibers to kind of latch on to each one of the chromosomes. Okay, homologous chromosomes do not associate. That'll make sense later. So remember, homologous means the same. So the same chromosomes do not associate. Okay, everyone good? All right, anaphase. The chromosomes are pulled apart. So the centromeres start pulling on those spindle fibers, and they rip the paired chromosomes apart. And they pull them towards the opposite sides of the poles. Okay, anaphase pulled apart. Is everyone good? And finally, telophase. Now there are two. Okay, so the chromosomes have reached the opposite poles of the cell, and they begin to unwind. Remember, back in, where was it? Prophase. Okay, in prophase, remember they thickened and shortened. Okay, now they're stretching out again. The spindle fibers that came from the centromeres are now disintegrated. The centrioles are replicating, so there's enough for both new cells. The nuclear membrane forms around the chromosomes, and that results in two daughter nuclei. So now the cell has two nuclei in it. They're exactly the same. And then the cell cleaves and it divides in half. Okay, questions there? All right. Okay, so meiosis. In meiosis, you have 4 daughter cells, which are produced. Each daughter cell has half of the chromosomes of the parents. Remember, he was talking about haploid cells. Okay, so half of the amount of chromosomes. There's 2 sets of cell division involved. So basically, we go through all of those phases, except for interphase, twice. So it always starts out in interphase, right? Interphase is the kind of just sitting chillaxing. So it won't go back to interphase in the middle, but it goes through prophase, metaphase, anaphase, telophase twice. Okay? And this creates the sperm and the egg cells. And all the cells are different. Okay? All the the DNA in the nucleus is different, right? That's why you're not your younger sister's twin. Okay? Because if all the genetic information in each sperm cell was the same, and each egg cell was the same, then you and your sisters would be twins. You and your brothers would be twins. Okay? All right, so meiosis 1. So the first stage, there's meiosis 1, there's meiosis 2. Okay? Meiosis 1 has two main purposes. It is the reduction and division, so it reduces the number of chromosomes in half, making the daughter cells haploid from a parent cell which is diploid. During meiosis 1, most of the genetic information recombines. Okay, so it rather than in mitosis where the genetic information is identical, it kind of switches all over. Okay, so in mitosis, okay, all of the genes on each one of the proteins, each one of the chromosomes are the same. Okay, in meiosis, they're going to be different. They're going to go to different places. And that's what makes you genetically different than your siblings. Okay, so for the phases, keep in mind that before meiosis begins at all, the DNA undergoes replication just like it did before it started mitosis. So when you first see the chromosomes in mitosis 1, or meiosis 1, they have their sister chromatid already, just like in mitosis. In meiosis 1, we will be talking about tetrads becoming visible, lining up, separating, and decondensing, rather than chromosomes like in mitosis. Finally, cytokinesis occurs. Remember, cytokinesis is where the cells cleave apart and you end up with your two daughter cells. All right. So, prophase 1. Just like in mitosis, during prophase, DNA condensation occurs. The nuclear envelope and nucleolus disappears, and the spindles start to form. The big difference is what's going on to the chromosomes themselves. As the DNA condensation proceeds, the chromosomes first become visible. They become visible as the tetrads. So the tetrads are visible during prophase. Okay. You good? All right. So metaphase 1. The tetrads line up at the equator, and the spindles have completely formed. So those are those spindle fibers. During prophase 1 and metaphase 1, genetic recombination is occurring. So take a look at the genetic. We don't have that. Keep in mind that it only happens when there are tetrads. So As anaphase 1 gets going, the genetic recombination is over. Okay, so the genetic recombination is where the genes are switching places. So in anaphase, the tetrads are pulled apart and the chromosomes with 2 chromatids move towards the poles. Okay, so just like in anaphase and mitosis, they're being pulled apart. She talked to me. Okay, good. All right, and then in telophase, remember telophase, now there are 2 chromosomes with 2 chromatid decondense, so they're spreading out, and the nuclear envelope reforms around them. So now each nucleus is haploid. Okay, keep in mind that it is not the number of chromatids per chromosome that determine whether a cell is diploid or haploid. It's the number of chromosomes that are paired that determines this. Okay, so right now there are 46 chromosomes in this cell, okay, that aren't paired. So there's 46 single chromosomes unpaired in the cell. All right, meiosis 2. Now we take those 46 chromosomes and we're going to split them in half. At the end of meiosis 1, each chromosome still had 2 chromatids. That is double the amount of DNA that a cell should have. So the entire reason for going through mitosis 2 is to reduce the amount of DNA back to normal. Basically to split the chromosome so that each daughter cell has only 1 chromatid per chromosome for normal, as in normal genetic content. So phases, as you read through the phases of meiosis 2, you will see it looks just like meiosis 1. It is really similar to mitosis, so keep that in mind. The only difference is that the 2 chromatid per chromosome are not necessarily identical due to the genetic recombination during meiosis 1. So basically all that's saying is meiosis 2, we have all the chromosomes, or meiosis 1, it's finished, all the chromosomes are split, they're now haploid, but the chromosomes are different, okay, because of that genetic recombination. Okay, so now we're going to go through meiosis 2, so they split again, so that there's 23 chromosomes, because remember you get 23 chromosomes from your mom, 23 chromosomes from your dad, which gives you the 26 chromosomes you need to be a human. Okay, everyone good? Prophase 2. So again, chromosomes with Two chromatid become visible as they condense. Okay, so they're squishing together. The nuclear envelope and the nuclei disappear, and the spindles are formed. Okay. Metaphase 2. So the prophase 2? Metaphase 2? We good? Okay. So the chromosomes with two chromatid line up along that equator, and the spindle is fully formed. Although genetic recombination primarily occurs during meiosis 1, the way chromosomes line up during metaphase 2 can also help to make unique daughter cells. Okay, finish copying this out and we'll go for a break. Okay, we did metaphase? Okay. Anaphase. Remember, pull apart. Anaphase, you pull apart. Chromosomes split so that a chromosome with only one chromatid heads towards each pole. So now the sex cells are almost to where they need to be considered a sex cell with only the 23 chromosomes. Okay, good? Last page. Telophase 2. Chromosomes with only one chromatid decondense and get surrounded by a new nuclear envelope. Okay, so this is finally where the nucleus is closing. Okay, the cells are starting to cleave using cytokinesis. And the four daughter cells are now all haploid and have the right amount of DNA. They are ready to develop into either sperm in a male or an egg in a female. Questions? Comments? Concerns?